this lecture and simply give you some idea in protein structure and prediction in 3D. Where are we at? Oops. So let's begin with a simple statement. Amino acid sequence determines 3D structure, established by Christian Anfinsen, that got him the Nobel Prize in literature and chemistry in 1972. And that implies that in principle, if the protein knows how to fold, we should be able to do the same thing. We should be able to predict protein structure from first principles. But it's sort of complicated because it's a many-body problem, and their computers are not fast enough, and the force fields are not known well enough. So it's complicated. But in terms of numbers, where are we standing today? Let's begin with a simplest statement. What fraction do we know experimental high resolution structures for? High resolution structures for me is X-ray crystallography in this context and NMR. We have 20,000 proteins in human, and for about 3,000 of those 20,000, there is some structural region known. So that does not mean that the structure is known for the entire protein. That means that there is a protein, a human protein, for which the three-dimensional structure is known. Why is that not the same thing? Anybody? To make this point that there is a high bias in the PDB toward shorter proteins, and what I'm talking about is not quite relevant for that. And the way the numbers that I'm showing here sort of translate to what it means for every single human protein. Again, about 15% of the human proteins are such that they will be covered by some experimental data point. When we look at UniProt, there is a number about 160K, so that is the entire PDB, and that is less than 1%. And I realize that I'm going to pause. There is a factor of 15, but the factor of 15 is a gross underestimate of the reality, because making a structure for a human protein is at least 10 times more difficult than a bacterial protein. Most of the proteins in UniProt, most of this 220 million, or 200 million, are bacterial proteins. So this number is highly enriched in bacterial, and in fact, in principle, this number should be higher than this number, because if that would be any eukaryote, that is 10 times lower, so that most likely would be 10 times lower than this number. So that shows you that the overrepresentation of human is even more extreme than the factor of 15 seems to indicate. Anyway, that gives you some idea where we are standing in terms of structure determination. And that gets me to the best way of predicting structure. Best in two ways: highest coverage and highest precision or accuracy. Comparative modeling, homology-based modeling, or homology-based inference. That simply comes from the fact I show 4 proteins here from bacterium, fly, and 2 from human. Essentially, they have very similar three-dimensional structures, although the bacteria and the human, here the first human, have only 19% sequence identity. Nevertheless, the structures are very, very similar, and that gets to the simple idea. If I have a query protein Q, I find a protein in the PDB for which I experimentally know the structure T, and if Q and T have enough sequence similarity to establish that they have a similar three-dimensional structure, then I simply say Q has the same structure as T. Again, this is an empirical way of determining the threshold. Once I make that statement, Q is similar to Q, I refine the alignment between target and template between Q and T, and I build a model. The simplest way of building a model is simply copying the coordinate of T to Q. So you simply take, for every particular position that is aligned between the two, you take the coordinates from T and copy them onto Q. That is the most covering, so that is the most of the protein structures predicted today are predicted this way, and that is also the most accurate structure prediction method, no matter what I'm going to say later. There are constraints, obviously. You need a template that fits above that threshold. It works only in the region where the two align, so not the entire Q is aligned. Not even if Q is aligned from here to there with T, there may be many regions in between where T is not having a structure that also fall out. It's not only the limitation in the region where the two are aligned, but beyond that. And you cannot do new discoveries. You can never discover new structures. This only repeats what you already have in the PDB, right? Those are the limitations. Now, overall, there's a model builder Swiss model that makes models available, and what I'm showing here is for mostly covering the human protein to covering only a small region. So mostly covering is 5,000, only a small region is 18,000. Now these two numbers here would, when you sort of do the addition of 3 plus 18, you have more than 20. And that again is this issue of multiple domains. So we have a sort of double counting here, because that could be a different region.
I have a question.
Shoot.
You said you can really not discover any new structures, but like
better than that. Anyway, Swiss model can model something that we could argue is sort of 1/4 to almost all human proteins, or in all human proteins, one fragment at least. So 100% here is still, due to the shortage of the PDB, would still be only roughly 1/3 of the human protein, 1/3 of the residues. So even if this number were 100%, this is still not 100% of human, it's only 30% of all, 40% of all human residues. I put the number for UniProt in gray simply because we assume that applying Swiss model, you can model 40 million UniProt proteins, which would be roughly 20% of UniProt, or in that fraction, but it does not exist as a data set. It does not exist as a database. This one does. So Swiss model has enough resources, or the developers of around Thorsten Schwede, they have enough resources in Basel to really make human and 10 other or 12 other organisms available, but they cannot do that for UniProt. Okay? That's why these numbers are in gray. So this actually exists, and this sort of could exist. And again, if you pick any of those proteins overnight, you can get a Swiss model prediction. And that gets us to something that does it even faster, and it's the server that was initiated by Sean O'Donoghue, and then Andrea Schafferhans joined the game, Aquaria. Aquaria is a thing that does visualization, is very good at visualization and modeling, but is not as accurate at the modeling as Swiss model, because essentially all it does, it says that I have the template from the PDB that I talked about. I have my query, and I say that the query fits to the template without refining. So Swiss model uses a lot of its computer resources to refine from the state of copy coordinates. What Aquaria does, it doesn't actually take the coordinates. It simply says, I have the 3D structure template here, and it's like copying the coordinates. I pretend that my query has simply the same thing, and I'm going to show you the structure of T put onto the sequence of Q. That essentially is what it does, and it provides you a lot of tools around that for visualization. Because they sort of do not do the detail of the modeling, they can do that on a larger scale. So they in fact provide Aquaria models for the entire Swiss model, and updates on at least a base of once a year. It is sort of a kit that allows you to do exploration of structure, or use structural knowledge to do a lot about your protein. That is essentially what is optimized on. That is a movie that shows it from Sean. It's really amazing what you can do about it. It is a great interface, and here is one way of applying Aquaria that was done last year, or over the last 12 months. Sean and Andrea have essentially run Aquaria through SARS-2 through Aquaria, and sort of put up an image of all the structural knowledge of SARS-2 plus resulting biological interpretation. As far as, so some of these proteins look like they mimic some other proteins, some other mechanism, some other sort of hijack pathways from the human host. So it's a great study showing how you can learn from structure. And that gets us into first principles, molecular dynamics. Protein folding should be doable from first principles, and that is essentially the idea behind molecular dynamics, which got the Nobel Prize in Chemistry in 2013 to Martin Karplus, Michael Levitt, Ari Rochelle, who were the ones who came up with this idea of essentially distributing or putting this problem into two parts. One where you do sort of more detailed classical physics, one where you do quantum mechanics that is using a lot of computer resources, the binding side, and one that is the environment, the water, in which you have just dielectric medium, so some average constant. So you have three different types of physics that come together in a model. In the binding side, the quantum mechanics. One aspect of it is you simplify by, rather than modeling atoms, you model residues as blurbs, and that you can in principle do on a computer, and you can do it to the level in which, starting from a three-dimensional structure in 1979, Michael Levitt generated this movie that he made available online, and has been making it available for many years for the people who care. This is the first movie of molecular dynamics, and as you see, sort of this is it looks like the movies from the olden days, and really is from the olden days. And that took, I can't remember, at some point he explained how long this took. It's a substantial amount of computer resources and other resources to produce this movie. And what you see is the bovine pancreatic trypsin inhibitor, BPTI. And one aspect that you see here is an immense amount of movement. And that gets us back to the point of structure. Structure is the background, is the theme, but it is also the mechanism, or is the backbone that allows motion. Function is motion. So this is not just molecular motion as you sort of expect it in random motion, but it's very controlled motion, and it's very much steered by the way the structure looks in detail. What you do see is also that in some regions you have much more motion than in others. So here you have, so for instance, one where the molecule can get in and sample spaces, right? You have regions where much more width of motion is the case. You have regions where clefts open and close. So the surprising reality here, seeing that movie for the first time, was that in fact the force field could capture the reality, and that this capturing allowed you to sort of explain something you see and predict some things that you don't see. Now, mind you, we are now 40 years later here. We can still not see this movie experimentally. So this is still something that you can only see on a computer. So you absolutely need molecular dynamics for that movie. And here is sort of a modern version from D. Shaw research on a machine that is way larger than the one Michael used a few years before that, called Anton 2. And this particular one is a spike, the trimer of spike, two proteins binding to some candidate for treating COVID with a drug, for some drug target. What drug, I don't know what the drug is. That is sort of provided by the D. Shaw research enterprise that built this machine, Anton 2, that is hardware and software optimized for molecular dynamics. We start in this movie, essentially from three-dimensionally known structures, and then do the molecular dynamics for those. Thereby they can really simulate long amount of time. But they can in fact now predict 3D structure for very small proteins. I believe this is an overestimate. So I said they can do it for every protein that is until 100 residues long. Is not quite true. If they could, this is sort of about 1K human protein. So roughly 5%. And for UniProt, that would lead to these numbers here. 5% would be something like 10 million in UniProt. But again, this requires, Anton 2 can do it, but it needs a lot of time and a lot of money to do that. So I'm not sure they will make something like that available. But this is been something that sort of has happened over the last few years to the level that we could essentially fold up at least some proteins with molecular dynamics, the short ones, and that we can do much more in terms of simulating binding drug interactions. So this is becoming part of what drug design is using. And that gets us to the field of AI, predicting three-dimensional structures. And I begin with David Baker, who is not at all representing AI. Essentially, the method, the suite of software for macromolecular modeling that David started, Rosetta Commons, is something that is sort of using MD comparative modeling, other aspects of structure prediction. The one that Yannick mentioned, so fragment assembly, was for many years the thing that Rosetta was brilliant at. And ultimately, if you want, in some sense, fragment assembly is something like comparative modeling. It's just that the template that you use is getting shorter. That allows you to find templates, find more templates, so to reduce the threshold in sequence identity, and then combine the model from different templates. And if you want to, in the framework of Rosetta, that evolved as a way to sort of use more generic or use more than one template, so become more complex, more advanced in doing comparative modeling and bringing comparative modeling into the world of free modeling. And that combination is something that the world of Rosetta started. Here is an early publication from 20 years ago from David Baker and André Charlie. André Charlie then was strongly developing Modeler, still developing Modeler, another comparative modeling method. Anyway, so that is Rosetta. The Rosetta went more into the AI direction. Zhangji Yang is one example for that. There's TR Rosetta. TR Rosetta is a method that allows you to predict protein structure on the web, is a web server that is available. And that web server essentially has all the elements of structure prediction that we talked about. It uses advanced methods of profile alignment, HH blitz, to build multiple sequence alignments, finds more homologous templates to do comparative modeling, uses these into machine learning to predict distance maps, uses those or uses some logic to refine those distance maps, and put those distance maps into some energy minimization that then brings us to the final model. If you remember the talk from Konstantin on Tuesday, essentially he began using TR Rosetta at this point. So he put his distance prediction, skipping the smoothing part here, directly into the energy minimization, and then he got the 3D structures out of it. So that's the part that you saw Konstantin present when he essentially predicted, showed the results of his prediction in 3D. And again, the major part, I'll get to that in a moment, of using the alignment information, the major part of that is really evolutionary couplings, and evolutionary couplings have a lot to do with having very huge alignments, and very huge alignments have a lot to do with doing something that is way beyond PsyBlast, HH blitz, profile alignments. And way beyond PsyBlast also implies this is not the sort of thing that you can do every day, every minute. So this is something that is limited to running a limited number of sequences. So if you work on a protein as an experimentalist for a long time, years, your life, then you can wait many days for a good alignment if you get something out of that. And this is what these kinds of techniques are made or optimized for. Now, TR Rosetta would essentially be able to predict for every single human protein. I am not sure to which extent TR Rosetta really is handling membrane proteins correctly. So this membrane proteins account for something like a fourth of all the human proteins. So this may be more accurate to say 15K, maybe more accurate to say 75%. But this 75% in contrast to these numbers here really is covering the entire length. So other than there are certain things TR Rosetta will not completely be able to handle, coil disorder, membrane region. Other than that, TR Rosetta in principle can predict the entire thing. It is still grayed out because this is not available as a data set, because they don't have enough resources for that. But in a collaboration with the European Bioinformatics Institute in Cambridgeshire, they generated a model of this quality here for every family in PFAM. That's about 15K, so the number is not that different. It is still slightly different in terms of job size because the PFAM is even shorter than the PDB. So the 20K human proteins are much longer, so there would be a larger job. But in principle, this is sort of feasible. Now, that gets me into a slightly different direction, which ultimately is, maybe I should turn that around, that's the beginning of it. But for me, this was the first time that I felt there's light at the end of the tunnel, and that's the work from Debbie Marks and Chris Sander. I have to update that. Chris is no longer at Sloan Kettering. Chris is also at Harvard now. And Thomas Hopf helped very much when they published the first paper that used protein sequence and predict membrane protein 3D structure directly from sequence using correlated mutations or evolutionary couplings. Konstantin talked about it. And this idea of correlated mutation evolutionary couplings is the crucial idea for being able to get good contact predictions or distance map predictions and to actually use those to predict three-dimensional structures. They put forward in a Cell publication in 2012 three predictions of three different membrane proteins that many groups had been working on for decades without being able to see this structure. And with the predictions for the first time being available, completely new structures, the experimental labs suddenly understood their data. That's why that was published in Cell, although it was sort of bioinformatics. And Cell, I don't know how much you know about that. Cell, for molecular biologists, cell biologists, Cell is sometimes more important as a publication than Nature, because it's just one of those very solid journals. So that was an immense achievement, and that immense achievement came because that was the first time. And underlying, again, I repeat, is evolutionary couplings, and evolutionary couplings are underlying any advance in AI and structure prediction, except for the one from Konstantin. That's the big exception. And now we get into the world of Google, DeepMind, AlphaFold2. Now, here's one way of picturing the achievement of AlphaFold2. So these are group names participating at CASP 14. These are just numbers, so even if I blew this up, you could still not read this, or I mean, it would still not give you a name. The score here is essentially how good they do, and you see that there's one group completely standing out from the entire field, and that one group is AlphaFold2. Now, another way is unfortunately more complicated, and that is this one here. On the y-axis, you have a score that measures how good a three-dimensional structure prediction is. Anything above 50 here is really sort of good. On the x-axis, you have target difficulty. What that means is essentially, if you have a query that has 98% sequence identity to protein in the PDB, then that's very easy. It's going to be here. If the protein has a structure that has never been seen before, then that's on the side of very difficult. If it is the kind of thing that Yannick asked, where you have sort of at least certain region of, I don't know, 30 residues that have some similarity to something that is already known, then that is sort of somewhere in between, okay? So the more difficult, the more different from anything we have seen before, the more to the right on the x-axis, the more similar to something that we already have in the PDB, the more to the left. Each curve is one of the 14 CASP meetings, and it represents for all the targets at that meeting. Typically there are in the ballpark of say 50 to 100 targets. For each one of these 50 to 100, they simply ask, how good was the prediction for the very best model? I don't care which method that comes from, but the very best that existed. I know the answer, so I can choose the best one. And you see that for the very difficult one, I said that 50 is sort of what you need to be. And you see for many CASPs, none, not even the best model really got to the very difficult targets. And that is changing just over the last 2 CASP meetings. And we are now getting to the very high point here. That is CASP 14. And green is CASP14 removing AlphaFold2. So if I would not allow AlphaFold2 to submit, and I would have to pick the best model from everything other than AlphaFold2, then that's the green line. So what you see is there's a big difference between green and black. So AlphaFold2, again, that's what I showed you before, made a big difference. Another thing that you see, even if I removed AlphaFold2 from CASP14, then CASP14 would still be better than CASP13, which is a red. So green is clearly higher than red. Okay, so there is an advantage, or there is the field advanced from CASP13 to CASP14 by some margin, and by a much higher margin from CASP12. And that ultimately, that much higher margin, in particular for the difficult ones here, that is all AI. That is all evolutionary couplings. That is all this part of having huge alignments. This jump here. Okay? That's the major thing that has happened. Where things are easy, not so much has happened. But where things are complicated, we move from clearly being not good enough to be useful to something that clearly is useful, and that jump happens at CASP13. And now the next point is that the advance from AlphaFold2 to the rest of the field is higher than certainly the difference between CASP14 and CASP13, but even almost as high for some targets. The movement from 12 to 13, 12 to 14 without AlphaFold2 is similar as the one from AlphaFold2 to the field. So it's just showing AlphaFold2 really is ahead of the pack. How come? And so we have said that before, and it's just important that I make that point here. We don't know. And this is not because the AlphaFold2 developers are trying to have this a secret. Not at all. That is the CASP process. And the CASP process has reasons for to discourage individuals from going out there and publishing their own method. Why is that discouraged? Because in the past some people presented at CASP and said, I'm the CASP winner. And the CASP organizers or the CASP assessors felt this is a lie, so they began shooting back at that. And rather encouraging that there's one common paper in which everybody who sort of was considered by the assessors as a CASP winner present their method. And that publication typically takes 1.5 years. So we expect that publication to come out ballpark summer 2020. And now there are, I believe, signs that AlphaFold2 will have a publication earlier than that, but not much earlier. I repeat, this is not because Google is sort of trying to put it under a lid. This is because of some process that evolved over the past and for which there are many good reasons. As it stands, all we can do is we can go by what has been published at the CASP meeting. Here's a link where, in fact, what I show you are cutouts from the slides presented. The talk, I'm not entirely sure whether it's public. Let's just put it like that. There are recordings of the talk. To which extent those are public, I don't know. I just participated in the meeting, so I saw the talk from John Jumper, and there were several other presentations of the AlphaFold2 team, or members of the AlphaFold2 team, and I'm sort of integrating some of those aspects. But those aspects are my understanding, and when you go to the internet you find many other ways of understanding. Sometimes they align and sometimes they are not quite aligning. Ultimately we can't say until this is really out. But essentially it begins with sequence alignments, just like everybody else, predicts aspects of distance maps, improves distance maps, and then does a structure prediction. So the first novelty is, in AlphaFold2, this is one end-to-end system. And that's a crucial point. The second thing is that, and that clearly is different from Yannick?
What do you mean with end-to-end system?
End-to-end system means that they are not developing independent AI components, and they're not using one MSA alignment tool, plugging that in and then do next step. So, but instead, there is one whole framework that does all the steps together. So all the molecular dynamic simulation here that comes at the end of the day is done by the AI system. And that essentially means you train an AI system in principle that optimizes from here to here. Every step is optimized. It's not quite as clear as I'm saying it. It's not quite as simple as I'm saying it. But that is, at least several aspects of the system are like that. And they have not, AlphaFold1 has not been like that. AlphaFold1 simply used sort of components here and there and combined them with AI. So now this whole thing is under the roof of one big AI. I suspect that there are still sort of independently trained, or it seems there are still independently trained components. So it's not quite end-to-end. So one aspect that is novel and important is that the alignments are done with larger databases. So they are not using UniProt. UniProt has 220 million sequences at this point. And what they use instead is a database called BFD. BFD has 2.5 billion. Now you may argue that 200 million versus 2.5 billion, okay, is a factor of 10. But it's actually not true, because BFD with 2.5 billion is actually sequence unique at 30%. And 30% uniqueness of UniProt is more in the ballpark of 40 million. So the factor is much larger between the two. So it's a huge database. And on that huge database, they do a genetic algorithm. This is sort of the slowest search you can do, but it's also the most detailed one, and it's important. It's a crucial point because of what I'm going to show you in the next step. Oh no, over next step. Let's make another step. I'm going to get on the next step for the importance of the genetic algorithm. Before that, there's a structure module. The structure module, essentially what it does, it sort of learns particular shapes. If you want, would bring us back again, Yannick, to your point of recognizing some local themes or some fragments, and then sort of thinking or consider or hoping that you can assemble those fragments. Ultimately, the important step is here, by recognizing those shapes, you have shapes that are recognized independently of the sequence. So in this step from here to here, you throw away the sequence. You have now a machine that has learned shapes it has seen before, and tries to sort of rearrange whatever prediction is in front of it according to the known shapes, completely independent of sequence. That is important because this sort of allows new discovery. This could essentially mean that it's exactly what you would sort of assume that a fragment assembly should solve. You have things that are similar in their shape, but they look very different in sequence. And you could rearrange shapes slightly differently. How important that is, we have no idea of telling. Clearly, this way has not been used in structural prediction before. And it seems that this was a major element. So they provided data in their statement. This was very crucial for their success. So they provided data where they showed what would happen if they wouldn't have done that. So there were several examples where the final result would not have been right. Now we get to another novelty, and that is the one that is for which the genetic algorithm is crucial. So there is an iteration. So you use the alignment as input to AI that predicts distance maps. Then you use the distance map to rearrange the alignment. And that is exactly, so using constraints from structures to refine your sequence alignment is exactly the kind of thing you can do with GA, with genetic algorithms. But you could not do that easily with the kinds of alignment methods that we talked about in this lecture. So you need a GA for that. And you need a GA because it's a mechanism in which you can easily put structural constraints. T-coffee is one that I briefly mentioned in our lecture, that in principle could exactly be used for something like that. What you see down here is the predicted distance map. There's no right or wrong, so this is the prediction and the observations. There's no observation data shown, it's all prediction here. Again, the distance map is symmetric, so the upper and the lower diagonal, typically what you see in methods in publications like the stuff from Konstantin, in the upper diagonal you have the observation, and the lower diagonal you have the prediction. In this case, it's both prediction. What you do see from step 1, 2, 3, 4 is that the map is refined. So one thing that you clearly see, you have almost no signal except for the diagonal here. Another thing that you see is that here you have essentially a lot of long-range contacts. Here this reduces, and here you have really something that looks more like a typical distance map of a typical protein. I'm not saying that I have any reason to believe this is more right than that on average, but it looks more correct, right? And this is all by iterating between creating an alignment, using that to predict distances, using the predicted distances to refine the alignment, using that to improve the blah, 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 blah. That's the cycle. That seems to be another crucial step. And again, they showed data how the structure prediction really improved iterating through this. And by the way, this is also one of the reasons why it's expensive. So it takes a lot of time. Now, can we catch up as a field with AlphaFold2? So here's a publication that I became aware of yesterday, but I actually sort of looked at it only just before the lecture today. So I cannot say that I understood it or that I have much to say. What is clear is this is the attempt to somehow use AI as presented by AlphaFold2 to catch up with AlphaFold2. The way it is described in this manuscript shown here in bioRxiv is essentially looking at some aspects of predicted by or introduced by AlphaFold2, combined with some other aspects of AI that Google at least didn't talk about as clearly as it is presented here. So the result then appears to be something that is a mix between what some people do in order to improve AI with what AlphaFold2 is doing to get something that they claim is, I have to look in more detail, but reaches AlphaFold2 within a margin of sort of 80% of AlphaFold2 or something like that, which would be a big improvement over what the entire field has been doing until December. And this being only 6 months thereafter, after December, and this involving many of the players who presented their tools in December, this would be clearly a step forward. Again, time is unclear how much of that is true, but what you see is how much AlphaFold2 is changing the field in terms of numbers. AlphaFold2 again, just like TR Rosetta, can predict structure essentially for every single protein in human, for essentially every single residue in human. Unlike TR Rosetta, for AlphaFold2 we know that it clearly can handle membrane proteins. We don't know whether it can handle all of them. We don't know whether it can handle domain boundaries. We don't know whether it can handle every residue in human. Clearly disorder, more likely not. But this is more likely to be closer to 100% than TR Rosetta. It is more gray or thinner in the way I show it, simply because the required computer resources are immense. And again, how in detail, how much is unclear, but there are statements out there that a single prediction is in the ballpark of 100K. There are statements out there is in the ballpark of 10K. Let's look at the 10K number. If it were true that this is in the ballpark of 10K, then this would still be in the ballpark of at least $200 billion, right? And $200 billion is a big number even for Google. So this is not easy to produce. DeepMind has announced that they consider making something like that available. There's no statement whether that will be freely available or not. We really don't know at this point. But this is, whether it is available, it will become available over the next 5 years or not, this is an immense opportunity. There's a big step. And maybe what I just showed you from this David Baker and many other PIs, so it's not only David Baker, there are many people here. Dick Grisham, for instance, is just one of them. There are many people on here who are experts, many groups on here who are experts in the field. Maybe this is going to sort of get close to it, and some of that will be available.